The thylacine, the Baiji, Spix's macaw. What do these species have in common? Well, besides the fact that they're all very cool and awesome species that have existed, all of these are species that have gone extinct, partially due to human interference. Yeah, go on. Feel bad about the actions of our species. We all deserve that guilt. And while we're out here thinking about how bad you feel about the deaths of these amazing species and the so many others that have been erased from this plane of existence forever, permanently, we're never getting them back. Sorry about that. Let me tell you about a little concept we the people like to call de-extinction. That's right, no longer are your Jurassic Park dreams a thing of fiction. I mean, well, technically they are because dinosaurs have been dead for tens of millions of years. Yeah, there's no getting them back. We still got that. We're bringing stuff back to life. That's how it goes. Okay, so by now you may be excited at the possibility of bringing the dead back to life. And I am sorry to let you know that, no, that's not what this video is going to be about. Yeah, I know, you're probably wondering, why would I tell you about all this when that's not what I'm going to be actually telling you about in this video? Well, dear viewer, listener, person, first reason is that I, as a person, am the type to go off on ridiculous tangents that occasionally don't even relate to the very first subject because I keep playing off of what I say next, and also because these subjects do happen to be related. Because guess what? Well, instead of using the technologies that we would use for the de-extinction process for, well, de-extinction, we can use it to stop species from becoming extinct in the first place. Okay, you're probably thinking, wait, what do you mean by that? Why would we use de-extinction technologies for something other than de-extinction? Well, let's get into it. Introduction, placeholder sounds. So before we really get into, well, everything, I should probably answer the question of, what is de-extinction and how does it work? Well, de-extinction, to put it quite simply, is the process of bringing an extinct species back to life. Pretty straightforward, right? I mean, it's all in the name. What else would de-extinction mean? Now, it's a fine concept, but how do we go about de-extincting, unextincting? De-extinction grammar can be a little weird, sorry. So how do we go about bringing these species back to life? One of the more straightforward ways, well, not really straightforward, but one of the more predictable ways to go about bringing the dead and gone back to life would be through the use of gene editing and, to that extent, cloning. Specifically, nuclear transfer and germ cell transfer. Nuclear transfer, to put it simply, involves taking the nucleus of a cell with a full set of genetic information and then implanting that nucleus into a nucleus-less egg cell, an egg cell that is not having a nucleus, so that it develops into an embryo and can be birthed by a surrogate mother. Okay, it's not that simple, but if it works, the newly birthed individual should be a clone of the individual whose DNA and nucleus was used. Textbook cloning. This method is one that has been in the forefront of de-extinction efforts recently, having been successful in cloning species such as the gastric brooding frogs, otherwise known as platypus frogs, and Przewalski's horse. Yeah, that's pronounced Przewalski. No, I don't know why it's spelled with a P. Germ cell cloning is a little bit different. It does involve the editing of the embryo, but like a little to the left and down. Specifically involves the editing and transfer of the primordial germ cells of the embryo. Primordial germ cells are the cells that develop into the various gametes, cells used in the process of reproduction, typically sperm and or egg cells. And by editing the germ cells of an individual, it is possible to directly affect the traits that their offspring will have. It's basically just the cloning, but with extra steps. You might be wondering, why use germ cell transfer when nuclear transfer is so much more straightforward? Well, that's simple. Y'all know birds, right? Okay, that was a dumb question. I'm pretty sure everybody knows birds, because they're just about everywhere. Just about everyone has probably seen a bird before. Anyways, you'll probably know that birds lay eggs, and that is what separates them from mammals. And the way that an egg is created and develops inside the bird makes it just about impossible to use nuclear transfer on birds. With germ cell transfer, it's just really, really hard to do. But there's still a chance. And that pretty much sums up how gene editing and cloning can be used in de-extinction. Another method that can be used is selective breeding for certain traits, but there could be some debate whether or not this is really bringing the species back to life, as the traits that are in the selectively bred individuals may not be the same sort of traits as the ones that were in the extinct individuals, and thus not really being that species anymore. Progress has already been made in the de-extinction efforts to date, such as the successful cloning of species such as the bucardo and, as mentioned before, the platypus frogs. Although successful is a relative term, as while the de-extincted individuals did have the genetic material of their respective species and were technically alive, none survived more than a couple days. Even more recently, a cloned Przewalski's horse, or P-horse as they are sometimes referred to, was born using frozen DNA from the San Diego's Frozen Zoo. More on that later. Of course, since de-extinction involves basically using science to defy the course of nature, there are some setbacks. They include various factors, such as the difficulty of actually finding and stringing together the DNA for certain extinct species, and finding suitable habitats for the recently unextinct species. But in the vein of the use of de-extinction for conservation purposes, a more relevant worry has to do with the possible lack of biodiversity that comes with a cloned/genetically engineered population. The genetic diversity of a population can act as a failsafe against certain less than optimal circumstances, such as a disease outbreak. In populations with a high level of genetic diversity, it's more likely that in the case of a disease outbreak, among other things, there's a higher chance for the population's survival. While some more vulnerable species do usually have a lower level of genetic diversity, it's not necessarily an indicator for the population being in danger. Like, it may end up surprising you to know that the primate with the lowest level of genetic diversity is actually humans, and we don't seem to have died out or gotten that close to it. Although, the disease thing, There's also the factor of finding surrogate parents for the individuals created through the use of de-extinction technologies. Obviously, we probably can't grow these guys in some test tube in a lab somewhere, so they would need to be incubated in another actual living body acting as the surrogate. So we would end up typically using a surrogate from the closest still living relative to the species that we are essentially resurrecting. But what if it just isn't possible for us to do that? What if even the closest relatives are incompatible with the birthing needs of the species we are bringing back? In order to successfully use de-extinction processes such as the cloning, we need to know how to circumvent these obstacles. Alrighty then, now that we're somewhat familiar with how de-extinction and the processes of that would work, allow me to introduce our featured species. I'm gonna show you guys a clip from a video here. This clip is from a video posted in 2009 by the BBC, British Broadcasting Corporation, which features a quite interesting individual of a bird species native to New Zealand. So, I'm gonna guess that a few of you are going to want a little more context for this, based on what you've seen, as it's not really that good of an introduction by itself. So this clip is a part of a video taken from the BBC series Last Chance to See, which aired in the United Kingdom from September to October in 2009. This clip is from the 5th episode of the series and is posted on the BBC YouTube channel with the title Shagged by a Rare Parrot. Yeah, I know. The episode is titled after the species of bird that is featured here, the kakapo. The kakapo, also known as the owl parrot, is a species of parrot that is native to the islands of New Zealand. A little side note, New Zealand has about 600 islands, which is pretty cool. Currently, the kakapo population consists of about 208 individuals, which has earned them the category of critically endangered on the IUCN Red List, which is probably the opposite of an achievement here, but, you know. So, how would we go about using de-extinction to help the kakapo? Well, first of all, in order to talk about what we can do, we should talk about what has been done. Since the 1950s, the government of New Zealand has made an effort to grow and preserve the population of kakapo that are still here today. Currently, the kakapo population is limited to 4 predator-free islands in the New Zealand archipelago, with the individuals that inhabit the islands being monitored closely. The reproductive practices of the kakapo population are also closely monitored, and I don't want to say manipulated, but they kind of are. With a population that small, inbreeding is a real risk for the kakapo population, especially since the result is typically an unsuccessful hatching. And that, it sucks, as kakapos already mate and reproduce so infrequently. Their mating season is sort of tied to the availability to large amounts of certain types of fruit, which they use to feed their children. So the solution that has been implemented is using artificial insemination to basically make sure that the offspring of any given bird has as little a chance of inbredness as possible. Seriously, they'll even use drones to override matings between individuals who are, quote-unquote, incompatible. It's a little weird, but all in the name of conservation, right? All of this has led to a growth in kakapo population, even though numbers are still nowhere near high enough to be secure about the survival of the species. So, that's all fine and dandy, but where could we fit de-extinction technology in to help with this? Well, artificial insemination requires those doing the inseminating... Oh, that's not fun to say. Anyways, in order to do that thing, they need to collect the material used for insemination from members of the kakapo population. I mean, without the actual genetic material, how do you make the child? Okay, side note, I'm trying to stay nonchalant about this, but this feels really gross to say, I'm sorry. So, while we're in that vein of discussion, let me bring your attention back to the video clip I just showed you. Well, not just, but recently. Featured in this clip is a kakapo, or owl parrot, named Sirocco. Sirocco is an interesting case among the kakapo, particularly for his preference for the company of humans, especially intimate company. That's gross to say. This particular quirk of Sirocco's makes it particularly hard for those involved with the conservation efforts to collect his genetic material for artificial insemination, and that also kind of sucks because without his traits in the population, there's less diversity. But what if we could use de-extinction technology such as germ cell transfer and all that other stuff to bring Sirocco's genetics into the population without using the typical genetic material that would be collected from him? Using germ cell transfer to create a clone of a bird in question needed for reproduction, or at least one carrying some of their genetic material, would hypothetically allow those traits to be introduced into the population, even if the bird is not actively participating in the reproduction process. And that's all fine and dandy, but we could also take it a step further. With germ cell transfer, we could potentially alter and add certain traits to those kakapo offspring to increase the chances of successful reproduction and the survival of offspring. Certain traits could be changed to, say, alter the reproductive cycle so that the mating cycle is less infrequent, or to make sure that most of the offspring actually can make it out of the egg and survive into adulthood. But wait, why would we use something made for bringing extinct animals back to life to keep them alive? Isn't that not what it's meant to be used for? Is something you may be wondering now. Well, before you get your britches in a twist over de-extinction being used in cases pre-extinction, allow me to reintroduce you to the Przewalski horse. These horses are the last technically wild horse species alive today, as their ancestors were never actually domesticated at all. Pea horses were once critically endangered and even went extinct in the wild, but thanks to conservation efforts, they have since returned to their natural habitat and are no longer on the brink of extinction. But that's not exactly the information you're looking for, so let me introduce you to Kurt. Kurt was born in August of 2020, and he is currently living in the San Diego Zoo, I believe. But right now, the relevant thing is that Kurt was created using the cloned genetic material of a Przewalski's horse kept in the Frozen Zoo, which is a database of genetic material that is kept by the San Diego Zoo. They did actually do this to introduce that genetic material to the population. And I am not currently aware of any complications with Kurt's health, so this is, as of my knowledge, a success in the terms of cloning for conservation. So in conclusion, the science of de-extinction has driven the development of many different technologies and practices that can be used to aid conservation efforts for species that are in need today. And while we probably cannot, and almost definitely should not, bring back species such as the dinosaurs, as we have imagined of doing so since Jurassic Park era, we can use these de-extinction technologies to make sure we never have to try to bring back their descendants. Thank you.